hundreds of different amino acids are observed in nature, but only 20 amino acids are abundantly found in proteins. And these 20 alpha amino acids differ from each other only in the identity of the side chain or the R group. So if you remember, what we had drawn on the previous slide or in the previous video was we had our carboxyl group. Then we have our alpha carbon. Okay, then we have the amino group, so that's why we're studying alpha amino acids. And we said, okay, the R group attached to the alpha carbon, depending on what the identity of the R group is, it's going to dictate what amino acid we're dealing with. And so you see some different R groups here. Okay, now here they're drawn as Fisher projections, but you can see that when the R group is a methyl group, we have alanine. When the R group is a CH2OH, it's serine. Now you notice that these are L amino acids, and if you go back to the chapter on carbohydrates, the naturally occurring carbohydrates were all D sugars, okay, so they were all based off of D-glyceraldehyde. Well, it turns out the naturally occurring amino acids are actually L amino acids, and so their stereochemistry is the same as L-glyceraldehyde. So that means the amino group is going to be on the left-hand side as you see here. And so let's take a look at the amino acids, the 20 naturally occurring amino acids that we are interested in. And we're going to start with amino acids with non-polar side chains. And you can see that for each amino acid, not only do we have the name and the structure, but they also have a 3-letter abbreviation and a 1-letter abbreviation. Now, you're not obliged to memorize the amino acids for this class, but it's not a bad idea to do that because many of you are going to want to take biochemistry and you will have to memorize the structure of all the amino acids for biochemistry. You'll also have to know the names. And of course, you're going to have to know the 3-letter abbreviation and the 1-letter abbreviation for biochemistry. So again, nonpolar side chains. So these, you can see these are all alkyl groups with the exception of a couple of aryl groups. So let's go through them. First one is the only achiral amino acid, is when we have a hydrogen as our R group, and of course there is a second hydrogen which is not shown here. That's glycine. The 3-letter abbreviation is GLY, the 1-letter abbreviation is G. I'm not going to go through all the abbreviations for these. Next, if you have a methyl group, that's alanine. When we have an isopropyl group, as is shown here, it's valine. Okay, you can see valine is repeated in this table. Then if you have an isobutyl group, so you can see this is an isobutyl group here highlighted in green, it's a leucine. And if we have a sec-butyl group, then it's isoleucine. And you can see the isoleucine is interesting because it has not only the chiral carbon at the alpha position, but you also have a second chiral carbon here. Next one is proline. And proline is the only amino acid that has a secondary amine. So you'll notice it's a secondary amine here. And the R group is tied to the nitrogen, which makes it kind of interesting. Okay, so proline. Then we have phenylalanine. So we have CH2 followed by an aromatic ring. So that's phenylalanine. So you just take alanine and you add an aromatic ring to it and you get phenylalanine. And then tryptophan, and sometimes students will say, well, why is tryptophan in the nonpolar section? Because you've got a nitrogen-hydrogen bond here. Isn't that a polar bond? And the answer is, yeah, that bond is polar, but it's nullified by the fact that you have this big, nonpolar portion here from the aromatic rings that are tethered together here. So tryptophan is does have a nonpolar side chain. I should also point out that this group here altogether, this system is actually called indole. So this thing here, so this molecule has a name, it's indole. So you could say it's an indole side chain. Sorry, not the best handwriting there. But anyhow, indole, if you want to memorize that, you might find that helpful. Okay, next we have amino acids with polar side chains. So again, these all have polar side chains. And you notice some commonalities here. First of all, 2 of them have hydroxyl groups. 2 of them are primary amides. Next, we have amino acids with polar side chains, and you can notice some common features here. So 3 of them have hydroxyl groups, which of course are polar. Then we have primary amides on 2 of them, and then you have a thiol on one of them. Okay, so if we go through them, some of these I've found easier to memorize than others. Okay, the first two are asparagine and glutamine, and asparagine has one methylene before you get to the primary amine, and glutamine has two. So two, like that. Then we have serine, which is CH2OH. We saw that earlier. Then we have threonine, which has another chiral carbon in it. So you have two chiral carbons in the molecule. Then you have tyrosine. So if you take phenylalanine, that would be phenylalanine. If you add a hydroxyl group in the para position, you get tyrosine. And then the last one is cysteine, which has a thiol in it. So it has CH2SH, okay, like that. So cysteine, so these are all polar side chains. So we've covered nonpolar and polar, and the last two groups are acidic side chains and basic side chains. You can see these are kind of shorter. So aspartic acid has one CH2 and then the carboxyl group. Glutamic acid has two CH2 or methylene groups, and then the carboxyl group for your carboxylic acid, so aspartic acid and glutamic acid. And then for the basic side chains, these are sometimes tougher to remember, but you've got arginine, histidine, and lysine. And so that covers all 20 amino acids. And one last thing I'll mention is you've probably noticed how close the structures are of asparagine and glutamine to aspartic acid and glutamic acid, right? Aspartic acid, if you replace the hydroxyl with NH2, right, then it becomes asparagine, and glutamic acid, if you change this to NH2, it becomes glutamine. So you can see how they're pretty close in structure, so that just maybe makes it a little bit easier to memorize because of that pattern. Now, something interesting about amino acids is that our bodies can make 10 of the 20 amino acids that we need, but 10 of them we can't make, okay? 10 of them we have to get from our diet, and we call those essential amino acids. Now, meat eaters can get all of their essential proteins from meat, fish, milk, and eggs. So if you eat a piece of chicken, it's going to have all 20 amino acids in it, whereas it says here in the slide that no single plant source contains all of the essential amino acids. I know that that's not true. Sometimes I'll have a student who will raise their hand and say, that's not true. Soy has all of the amino acids in it. So I know that's not entirely true, but vegetarians do have to be a little bit more careful maybe about their diet to make sure they get the right combination of food so that they get all the necessary amino acids. Now, in addition to the 20 naturally occurring amino acids found in proteins, there are a whole host of other amino acids used by organisms for a variety of functions. If you take a look at the structures of GABA, so gamma-aminobutyric acid, and thyroxine, both of these are considered to be amino acids, and you can see why. The amino groups are highlighted in red, the acid groups are highlighted in blue, and GABA acts as a neurotransmitter in the brain, while thyroxine is found in the thyroid gland and acts as a hormone. And there are other examples of amino acids as well, but again, we're going to focus on the 20 naturally occurring amino acids found in proteins in this chapter. When an amino acid is dissolved in a solution at a pH of 1, both functional groups exist primarily in their protonated form. So if we take our amino acid and we put it in a solution, let's say, something like a pH of around 1, okay, then our carboxyl group is going to be protonated, and also our amine is going to be protonated. And so you can see in this case, overall, we have a charge of +1. And each of the highlighted protons has its own unique pKa value. Normally we call them pKa1, which refers to the pKa of the carboxyl proton, and pKa2, which refers to the pKa of the protonated amine. Now you notice that the carboxylic acid group is deprotonated first. That is, pKa1, that value refers to the acidity of the carboxylic acid group, while the second pKa, pKa2, again, refers to the acidity of the ammonium group. Some amino acids have side chains, whether they're basic or acidic, and those amino acids have a third pKa value associated with the side chain, And you can see some of the pKa values for the side chains shown here in this table. Now you probably notice that the pKa values for the carboxylic acid groups are in the range of 2 to 3 for nearly all the amino acids shown. For example, if we take a look at alanine, which is shown back here, you can see the pKa of our carboxyl group is 2.34. And that value indicates that the uncharged form, okay, the carboxyl group, and the anionic form, the carboxylate, are going to be present in equal amounts at a pH of 2.34. So if we have any pH below 2.34, meaning more acidic, the uncharged form is going to predominate. It's going to be protonated, whereas at any pH above 2.34, the carboxylate ion will predominate. And so what form is going to predominate at physiological pH? Well, again, and I'm repeating myself, we see that the pKa value of the carboxyl group in alanine happens to be 2.34. Okay, so for physiological pH, which is a pH of around 7.35 to 7.45, so we'll just put 7.4. Well, the pKa is 2.34, and physiological pH is going to be more basic. And so therefore, the carboxylate form is going to predominate at physiological pH. Now, when thinking about the pKa values for the ammonium groups of the amino acids, what the tables have shown us is that these values are in the range of 9 to 10 for nearly all amino acids. For example, if we think of alanine, it has a pKa2 of 9.69. And what that number means, that 9.69 indicates that the uncharged and the cationic forms of the ammonium group are going to be present in equal amounts at a pH of exactly 9.69. Any pH above 9.69, the uncharged form is going to predominate because you're in a more basic environment. But any pH below 9.69, the cationic ammonium group is going to predominate because you're in a more acidic environment. And so if you're wondering what's the point of all that, the summary is this, okay? At physiological pH, which again is around 7.4, amino acids are going to be zwitterionic, meaning the carboxyl group is going to be deprotonated. You're going to have a carboxylate, and the ammonium group is going to have that positive charge. So we have a zwitterion, right? Zwitterion means we have both a positive and a negative charge existing in the same molecule. And since amino acids are zwitterions, which is a net neutral compound, but it exhibits charge separation of the positive and negative charge, an amino acid can be thought of as an internal salt, right? You have the cation and the anion, and it exhibits a lot of the same physical properties as salts. For example, amino acids are very soluble in water, and they have high melting points, just like inorganic salts. Something else that's interesting about amino acids is that they're amphoteric because they can react as either an acid or a base depending on the situation. So if you take an amino acid and you treat it with a base, like a hydroxide shown here, the amino acid is going to function as an acid by donating a proton from your protonated amine, like that. However, if it's put in an acidic environment, right, you have hydronium ions here, in that case, it's going to protonate the carboxylate group to give you a carboxyl group, as is shown here. And so these two reactions show that in its zwitterionic form, an amino acid can function as either an acid or a base. For each amino acid, there's a specific pH at which the concentration of the zwitterionic form reaches its maximum value, and we call this the isoelectric point, or the PI. So isoelectric point, you definitely need to know that. And each amino acid has its own unique isoelectric point. For amino acids that don't have an acidic or basic side chain, so that would mean the polar groups or the nonpolar groups, the isoelectric point is just the average of the two pKa values, pKa1 and pKa2. So if you take a look at the example of alanine, which is shown down here, if you want to calculate the isoelectric point, you just take the average of the two pKa's. So again, just add them up, divide by 2, and you get 6.02. So that would mean at a pH of exactly 6.02, alanine is going to have its maximum amount of the zwitterionic form, where the carboxylate, or the carboxyl group, is deprotonated, and the amine is going to be protonated. However, if we have amino acids that have acidic or basic side chains, The isoelectric point is just the average of the two pKa values that correspond to the similar groups. For example, if we look at lysine, which has a basic side group, what we do is we take the average of the two pKa's of the two basic groups. So 8.95 and 10.53, we take the average of those. And so the isoelectric point would be 9.74. Whereas if you have something, an acidic side chain like you do in glutamic acid shown here, what you do is you take the average of the two acidic group pKa's, okay? And that would give you the isoelectric point. Amino acids can be separated from each other by a variety of techniques, and one of the techniques is called electrophoresis, which relies on differences in isoelectric values and can be used to determine the number of different amino acids that you have in a mixture. So what you do is you take a few drops of your mixture and you apply it to a filter paper or a gel, and then it's put in a buffered solution between two electrodes, a cathode and an anode. And then when an electric field is applied, the amino acids are going to separate based on their different isoelectric values. If the isoelectric point of an amino acid is greater than the pH of the solution, the amino acid is going to exist in a form that bears a positive charge, and it's going to migrate towards the cathode. And so if we think about lysine, which has a basic side chain, it has an isoelectric point of 9.74. If you put it in a solution that's buffered at a pH of 6, well, the pH of 6 is more acidic, therefore it's going to be protonated, right? So it's going to have a positive charge overall, and it's going to migrate towards the cathode. On the other hand, if you have something like glutamic acid, which has an acidic side chain, okay, it has an isoelectric point of 3.22. So again, if you put it in that same buffered solution at a pH of 6, the buffered solution is more basic than the isoelectric point, and so it's going to exist in its deprotonated form, which would be negative, and so it's going to migrate towards the anode. So again, just repeating myself here, the lysine itself is going to be positively charged, and the glutamic acid itself is going to be negatively charged. And so positive is attracted to negative, and negative is attracted to positive. Now, if you have something like alanine, so that's going to predominate in its zwitterionic form, so it's going to be uncharged. And so it's just going to kind of sit here in the middle. So you can see how you can separate these compounds based on their isoelectric points. Now, if you have two amino acids that have very similar isoelectric values, like glycine and leucine, for example, the amino acid with the larger mass is going to move more slowly because the charge has to carry a greater mass. Now, electrophoresis is an analytical scale technique. That means it can't be used to separate large amounts of compounds. Another thing about amino acids is that they're colorless. And so a detection technique is necessary in order to be able to see them or see the locations of various spots. And the most common method involves treating the filter paper or the gel with a solution containing ninhydrin, followed by heating in an oven. Because ninhydrin, and you can see the structure of ninhydrin, this interesting compound is shown here, it reacts with the amino acids to produce this purple colored product. So maybe I should try to find a purple highlighter here. Okay, that's close enough. Okay, so ninhydrin reacts to form this purple colored product, and then you can see that the nitrogen of the amino acid is incorporated into there, and then you get some other side products, like you get water, carbon dioxide, and this is an aldehyde here, so those are the byproducts. So when you stain amino acids, you get a purple compound regardless of the identity of the amino acid, as long as it's primary. And so that means that proline, which has a secondary amine in it, doesn't react with the ninhydrin. But the number of purple spots will indicate the different number of kinds of amino acids present that you have in your mixture. Question 25.1, although most naturally occurring proteins are made up of only L amino acids. So remember that the naturally occurring sugars are the D sugars, but the naturally occurring amino acids are the L amino acids. Made up of only L amino acids, proteins isolated from bacteria will sometimes contain D amino acids. Draw the Fisher projections of D alanine and D valine. In each case, assign the configuration as R or S at the chiral center. So if you remember when we talked about the naturally occurring amino acids, the L amino acids, if you want to draw a template for an L amino acid, or if you want to draw a Fisher projection, I should say, we always draw them like this, okay, where we have the amino group on the left-hand side We have our carboxyl group at the top, hydrogen over here, unless it's proline, and then your R group is going to be down like this. And so if we're drawing D-alanine and D-valine, we're going to switch it around at our chiral center. So we've got D-alanine, and so we're going to have our carboxyl group is still going to be at the top like this, but now our amino group is going to be on the right-hand side, just like D-glycine. The hydroxyl is on the right-hand side of the chiral carbon. And then alanine, the R group, is a methyl group. And then if we have D-valine, so D-valine, the R group in this case is an isopropyl group. So we can just take this guy, let's do a little copy-paste magic here. So we'll paste this in, and all we have to do to modify this structure to go from alanine to valine is we put an isopropyl group here. And then what is the stereochemical configuration? Well, both of these, you can see they have the same order of priority, that the first priority group is going to be this one, the nitrogen followed by the carbon attached to 3 hydrogens, followed by this carbon down here. And keep in mind that the hydrogen is coming out on a wedge. So whatever our configuration is, we're going to have to reverse it. So we're going from 1 to 2 to 3, which is counterclockwise, which would be S. But remember, we have to switch that. So the stereochemical configuration here is R. And for the same reason here, going from 1 to 2 to 3, we go counterclockwise, which would be S, but we switch it. And so this would be an R compound as well. Okay, draw the form of the amino acid that is expected to predominate at the stated pH. So in our textbook, this is question 25.4. So 25.4. So we've got alanine at a pH of 10. The carboxyl group is a pK of 2.34. The ammonium has a pK of 9.69. So we're at a pH that's more basic. And so therefore, the carboxyl group is going to be deprotonated. And we're at a pH that's higher than the pH of the ammonium. And so it's going to be deprotonated as well because we're in a more basic environment. And so the form that's going to predominate is going to be this form where the carboxyl group is deprotonated and our ammonium is also going to be deprotonated like that. So let me just move that up there. Okay, now we've got proline at a pH of 10. So a pH of 10 is going to be more basic on the than the carboxyl pKa, and so the carboxyl group is going to be deprotonated, but it's more acidic than the ammonium. And notice here that I put NH2 plus because proline is a secondary amine, and so our amine is going to be protonated, but our carboxyl is going to be deprotonated. So we'll scribble that down. It's going to look like this. So our amine is going to be in its protonated form like this, and our carboxyl group is going to be deprotonated. Okay, and finally we have glutamic acid at a pH of 3, and so a pH of 3 is going to be more basic than the carboxyl group, and so that's going to be deprotonated, but it's more acidic than the side chain and the ammonium, and so both of those are going to be protonated. And so we'll start by drawing out the side chain, which again is going to be protonated. And then we have the amino group, which will be protonated as well. But the alpha carboxyl group is going to be a carboxyly. It will be deprotonated. Okay, 25.7, use the data in the table to calculate the isoelectric point of the following amino acids. So we start with aspartic acid, which has 2 carboxylic acid groups. And so the isoelectric point of aspartic acid is going to be calculated by using the pKa values of the 2 carboxylic acid groups. And so again, you can go to the table and look these up. So we have 1.88 for the first carboxyl group. The side chain has a pK of 3.65. So we take the average of those and we get 2.77. The next one is leucine, which does not have an acidic side chain or a basic side chain. So the isoelectric point of leucine is just going to be calculated as the average of the pK of the carboxylic acid group and the ammonium group. And so I've gone ahead and looked those up as well. And so for our carboxyl group, it's 2.36. And for the protonated amine or the ammonium, it's 9.60. So we take the average of those and we get 5.98. And then lastly, for lysine, it has two ammonium groups. It's got a basic side chain. So we get the isoelectric point of lysine by taking the average of the pK values of the two ammonium groups. And again, I've gone ahead and looked both of those up. So we have 8.95 and we have 10.53. So if you add those up, divide by 2, we get 9.74.